Cell physiology, graded action potentials. Graded and action potentials. All right, so cells contrive simple separation of charges, but great things result. My haikus amuse me. At least have fun with that. All right, before we dive in to how, so the next, let's say this, the next several lectures are going to require an understanding of what are called excitable tissues. Excitable tissues. Yesterday, I introduced four tissue types, right? Four general categories of tissues. And what were those categories? I talked about two of them yesterday. Yeah, that's what I talked about yesterday. And what were the other two? Skeletal, muscular. Yeah, muscles. So, yeah. Jeez. I can. Thank you for doing that. Yeah, I'm PowerPoint and all the software that I'm using here is undoing me for some reason. It's not working well in this classroom. It's this system over here, I think. How about that? Does that work? Okay. Thank you very much. So for tissue types, yesterday I talked about epithelium and connective tissue, and then I ended by talking about how they form tissue membranes. So a tissue membrane is epithelium on top of connective tissue. Well, the remainder of the tissues are excitable tissues, neurons, nervous tissue, and muscle tissue. Both of them are what are called excitable tissue. Today, I'm going to set the groundwork for understanding what excitable tissue even means. All right, so we're going to talk about the basic cellular physiology that enables a tissue to be excitable. Okay, and before we do that, I'm just going to go through a little bit of basic cellular anatomy here in the neuron. All right, so a neuron has four regions, four general regions. There are the dendrites. The dendrites are how information comes into a neuron. In some ways, a neuron is a model for the way the entire nervous system works. The whole nervous system has sensory input or afferent input, processing, and then output, right? Or motor neurons. And a single neuron is the same way. So dendrites are the input. And then there is the cell body, which is made up of two parts, the nucleus and the perikaryon. I remember that was the joke. When I first started teaching anatomy in Texas, I was at A&M in Texas, and Rick Perry was the governor, and that guy, while we were down there, there was a story about how he was driving down the freeway, and he like saw a bunch of coyotes and took an, like, I don't know, he's like a Texas guy. Bam, 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 bam. He like pulled over and shot a bunch of coyotes and left them on the side of the road. Kind of outrageous to me. I understand. But yeah, I don't remember exact wording, but it was some joke about perikaryon. A different spelling of carrion. But if it helps you remember how to pronounce that word, perikaryon. Peri actually means beside, and karyon is a reference to the nucleus or the origin of the cell. The word karyon has a Greek root that means origin. So the perikaryon is the cytoplasm surrounding the nucleus. The perikaryon itself is important in terms of information processing within the cell itself. Information processing within the cell. We have, and we'll talk about this, the summative addition of excitatory and inhibitory electric fields in the neuron happens in the perikaryon. And then there's the output. There is the axon with a special place called the axon hillock. Hillock is just a little hill, a little bump. The axon hillock leading to the axon, which terminates in the telodendria. Telodendria, which have these little synaptic boutons on the end, or synaptic knobs, or bouton. All right, so those are the structural components of the neuron. Within a neuron, there are five different membrane processes that are going to happen, or that we can describe. The first is just the process of nothing happening. That is the resting transmembrane potential. And by potential, I mean electrical potential. Electrical potential. I'm going to describe if you're not sure what an electrical potential is, that's okay. I'll describe it a bit more in a slide or two here. But we're going to represent it with the letter V for voltage, because voltage is the electrical potential. It's the term that we use to describe electrical potential, voltage. And V sub R for resting. The resting potential is V sub R. So I'm going to go through resting potential and how the cell establishes that. Then I'm going to talk about graded potentials. And graded potentials are temporary, localized perturbations of the resting potential. So we're going to move away from the resting potential to a different potential. And it's going to be graded, meaning that there's a gradient to it that will drop off as you move away from the site of stimulus. So a graded potential, it gets stimulated by something, and we'll talk about what can stimulate that. And it reduces with distance from the site of that stimulus on the cell. All right, graded potentials. The third is the almighty action potential. So an action potential is also a perturbation in the resting potential of the membrane. But unlike a graded potential, it doesn't follow a gradient. It doesn't get less with distance. In fact, there are mechanisms to propagate that perturbation in the membrane potential uniformly down the length of that membrane. All right? It is an electrical impulse. It gets sent down the length of that axon, and it is potentiated, the stimulus that starts it's potentiated by a series of graded potentials, or perhaps just a single graded potential that is of adequate strength to trigger an action potential. They're triggered. They get triggered, and then they get sent along the axon towards the telodendria. Action potentials represent the output of a cell. They're the output of a cell. You guys following these ideas here? All right, so then the action potential impinges upon the synaptic cleft, the end of the axon, the telodendria, and then ultimately the synaptic bouton with its synaptic cleft. When I say synaptic cleft, I'm talking about the gap between the synaptic bouton of the signaling axon and the next axon down the chain. The cleft is the space between the two of them. That's where all the magic happens. And a synapse, or synaptic activity, is simply the release of neurotransmitter from the end of that synaptic terminal into the synaptic cleft, and it's going to stimulate a graded potential on the other side, on the receiving neuron, on the next neuron down the chain. All right, so action potentials cause a synapse to release the presynaptic terminal to release neurotransmitters, and it's going to affect the membrane on the other side of the synapse. It's going to cause a graded potential in the next neuron. And then finally, a little bit out of order here, really, I should tuck information processing between graded potential and action potential in this list. But I put it at the end because I talk about it at the end, and I may not even talk about it today because just handling the first four is sophisticated enough. And frankly, information processing would probably require me to have like 2 or 3, 4, maybe a whole class course on how these cells really process all the electrical field information. But what it is how a neuron responds. How a neuron responds to the different information that it gathers as represented by graded potentials. This is the integration of different stimuli. Sometimes the stimuli may be additive, and sometimes the stimuli may be contradictory. All right? Imagine, for example, being the president of the United States, and she is listening to all of her advisors. One person is telling her, do this thing, and another person is telling her, do that. And that individual is integrating those things, conflicting stimuli. Or perhaps there's a chorus of people saying, stay in the Paris Climate Accord, or something like that, right? It's information processing. Information processing. Okay. So what you see here is a motor neuron in black showing up. It's a motor neuron in black with a cluster of 4 different telodendria synapsing on a muscle fiber. These are each of those red bands, those wide red bands are muscle fibers, skeletal muscle fibers. So this represents both sorts of excitable tissue. The point of this slide is simply to say that the function, as it says in the bottom there, function of excitable tissues, neuromuscular tissues, depends on cellular membrane potential dynamics. So there is a potential difference, a resting potential difference that a cell establishes across the membrane. You can imagine the cell membrane being like a battery. Like a 9-volt battery, you have the plus and the minus terminals. You can imagine the plus terminal being outside the cell and the minus terminal being inside the cell, and there's a potential difference between them. So whatever these excitable tissues are going to do is dependent upon changes in that membrane potential, that transmembrane potential. So we call that dynamics. The changes are dynamics that happen in that cellular membrane potential. Everyone with me? Okay. So first, just a little bit of discussion about what these membranes are made of. Most of you, I actually don't know what they cover at 164, frankly, but in biology. But I'm not going to assume that they talked about what plasma membranes actually look like. I think they do, but essentially, a plasma membrane, the membrane of a cell, is a lipid bilayer. It's a lipid bilayer. You have a lipid itself is what's called an amphipathic molecule. Amphipathic just means there's 2 types of chemical properties in the same molecule. Half of it's going to be nonpolar, the greasy long lipid fat chain on the end, and the other is going to be a polar head group. Polar meaning it wants to be dissolved in water. It likes to polarize itself. It's dissolved in water easily. And so these lipids line themselves up in a bilayer, where on the outer layer we have the negative charges all lined up facing the water because they don't mind being in the context of water. And the greasy tails are all lined up next to each other because they are like and pointing to the inside of the membrane. On the other side, the inner part of the membrane, there's a similar layer of lipids that are all lined up. So we get this lipid bilayer, where there's a greasy interior and a polar surface on both the inner and outer leaves of the lipid bilayer. Is that review for most of you guys? Good. Embedded in the lipid bilayer are these transmembrane proteins. Transmembrane proteins. And how do we know that? Well, here's one of the earliest freeze fracture micrographs where they just took a tissue sample, they stuck it in minus 80 degree liquid nitrogen, and then they smacked it with a hammer. And they fractured it in such a way that sometimes the fracture splits along the membrane, the lipid bilayer. So you peel one of the leaves of the lipid bilayer off. And you can see, so this would be the surface of an intact bilayer, and this is a bilayer that has had the top ripped off, and you can see these little bumps here are proteins that have been embedded in this lipid bilayer. All right, so we can actually visualize that. Okay. So because a membrane is a cellular membrane, a plasma membrane, is made up of lipids, it's going to inhibit the passage of polar water-soluble solutes into the cell readily, right? Because say you have some sort of, whatever this thing is, it likes to be dissolved in water. It can't just pass through here because this polar molecule doesn't want to be in the nonpolar region between the two leaves of the plasma membrane. So these proteins, these transmembrane proteins that get embedded in the cellular membrane, they act as channels. They act as a way for solutes to actually pass across that lipid membrane. And in terms of the kinds of proteins that are embedded in a membrane that facilitate the transport of different solutes, there are two types. There are channels and there are carriers. So a channel is just a pore. It's just a hole. It's just as the name would suggest, just a passageway. Whereas a carrier is something that has a specificity to it. It's going to actively somehow interact. It's going to interact with the molecule as that molecule passes. So it's like having a door, an open door, versus perhaps having a bouncer or a doorman at the door to say who can come through and who cannot. Perhaps that is an analogy. Everybody with it so far? It's all pretty basic. So what are the things that dictate whether something's going to go through it or not? Well, two things. First of all, what's the concentration gradient? Is there some kind of gradient, right? So imagine we had 200 people in the hall. We have all this room out here. There's a high gradient, and eventually all those people in the hall, some of them would just come in here and equilibrate. Nature abhors a vacuum. Okay? But gating, specific gating, also is going to dictate what's able to pass through a channel or a carrier. And there's different types of gating. And here are some examples I have. Here's a chemically gated channel. So here's some solute, some positive charge that wants to pass across the membrane. And it can't because it's closed. But here's this chemical, ACH stands for acetylcholine. That's a neurotransmitter that opens up cholinergic receptors, which are on your muscles, for example. And acetylcholine will bind, and it opens this gate. And now suddenly this positive charge, whatever that is allowed to pass through that. That's a chemically gated channel. Here's an example of a voltage-gated channel. So this thing here, this little box, is called a patch clamp. Is anybody here planning on taking any classes with Dr. Martin, Josh Martin? Any of the any neuro students here at all? No? Well, he uses these. But a patch clamp is basically a voltmeter. Like an electrician would use. They'd stick in the socket to a wall and see what the voltage is. It's the same thing here. There's a patch on the outside, and then they stick a probe, a patch clamp on the outside, and then they stick a probe through, and they can measure the potential difference from the inside to the outside of the membrane. All right? And here at the resting potential, this gate is closed. The ion can't get through. The voltage that you see is 70 millivolts. In fact, it's negative 70. Negative just as convention, right? You can have positive voltage or negative voltage, and just the way the convention that they use to describe the membrane dynamics is described as negative. So we change that for some other reason. For some reason, maybe there's a graded potential somewhere, or some reason this membrane potential changes. We're not at resting potential, and this gate is sensitive to that. Maybe there's an amino acid that is going to get titrated somehow. It's an acidic amino acid. It gets titrated. The proton comes off or something like that. Or maybe it's a basic amino acid and it gets titrated. And there's a charge repulsion and it snaps that gate open. For whatever the mechanism, the change in the potential of this membrane snaps the gate open and the charges can pass through. All right? And we get to a different voltage, and you can have two doors. You can have a bimodal gate. Doesn't have to be, but the example I'm showing is. There's an activation gate and there's a deactivation gate, and they work at different voltages. Yeah, Mary. No, you're just waving at me. Hi. And then finally is a mechanically gated channel. Mechanically gated channels, what would be an instance? So a mechanically gated channel, let me say this. You have a deformation in the membrane somehow, physical deformation in the membrane, which opens up that channel and allows a solute to pass through. What would be an instance might you imagine if you were going to guess, if you were going to design a human, and these are the tools that you have to do it with, what instant might you use that kind of a channel in? Yeah.
Sorry.
Okay, so you're saying when a muscle cell contracts, there's going to be a mechanical deformation that's going to cause an action potential. So is that what you're saying? No, what are you saying? Tell me.
Well, you stated it.
Yeah, I mean, it was a good thought, though. You're thinking the right way about things that, actual physical stuff. But you were putting the horse or the cart before the horse, unfortunately. Anyone else? Think of something. Yeah, John. When you touch something. Thank you. Was my hand rubbing a clue? That's right. Sensory receptors. When you push on skin, for example, you feel it. And what's really happening, you're deforming the plasma membranes of those cells and allowing that cell to register that something's happening. Something touched you. Something changed the shape of that cell. In fact, these mechanically gated receptors are extremely important in all kinds of sensory receptors. In fact, your ability to hear is based upon vibrations in hair cells in your inner ear that are mechanically gated channels that they open up and close transiently based upon the vibration of those hair cells. We'll see that next week. Okay. So these channels can have different states. They can be closed but able to open. They can be open and activated, or they can be closed and deactivated. These are possible states for channels. I'm just giving you the range, the diversity of channels that are out there. All right. So I already said that carrier proteins interact with the molecule being moved. A couple things. Since they're interacting with it, they can become saturated. Meaning, say you had, has anybody gone to, I'm sure many of you have gone to a crowded, popular sporting event or a concert, or even just at the dining hall during lunch rush or something like that. And to get in, somebody's swiping your card or taking your ticket or whatever, and the rate at which people can go in is limited by how fast the ticket taker or the card swiper can handle you, right? They are saturated. There's only a certain speed at which they can allow people to pass in, all right? So they're saturatable. Carrier proteins are saturatable. There may be way more solutes that want to come in than that carrier protein can handle. And if that's the case, it's been saturated. And then there's another feature. These carrier proteins can either be active or passive. So if it's passive, that means that some kind of gradient, maybe a chemical gradient, maybe an electrical gradient, probably both, is going to be driving whether something is going across, or they can be active. And if they are active, that means they require energy. It's like they need a motor. They got to plug in to be able to push something across. And that requires ATP, which means those proteins are called ATP aces. So that's ASES, not ACES, but these ATP aces are ace at cleaving ATP. So ATP is right here, adenosine triphosphate, and this is one of the nucleotides, and then it's attached to these three phosphate groups. What happens is they cleave off one of these phosphate groups. They break this phosphodiester bond, which is a high energy bond, and that energy that gets released by cleaving off a phosphate group from ATP, adenosine 1,2,3 triphosphate, to adenosine 1,2 diphosphate. That energy gets transferred into the process of transporting the molecule across the membrane. Sometimes you want to transport stuff up a gradient. That takes energy, right? It's like maybe there was a zillion people out in the hall, but I need to put a couple more of you, and so I'm like pushing. I have to push you out into the hall, right? It takes some energy for me to do that. So any protein that has ASE on the end of it is an enzyme that is going to be cleaving something, or some kind of protein that some kind of an enzyme. It's an enzyme. It just means a protein is an enzyme. All right. So here, briefly, I probably should have cut this one out because it's not that important. But these carrier proteins can. Here's some more variations on the theme of what they can look like. They can be uniporter. It means they can transport just one thing in one direction, either in or out. It doesn't matter. I have it going in here. But a uniporter is going in one direction, one thing. You can have symporters, two things coming in the same direction. Perhaps one of those things is going down its concentration gradient. And if it's going down its concentration gradient, we're getting a release of entropic energy, right? We're getting a release of entropic energy. So we're gaining energy. And then that energy is being used to offset something that's going up its gradient. So we're getting a decrease in entropy. And this is one way symporters are one way to drive transport of a molecule of interest using entropy. And this doesn't have to be in the same direction. They can be going in opposite directions. All right. So there is an electrical gradient across the membrane. I guess I've already said this since the beginning of the hour here, but we get a separation of charges across the membrane, and this transmembrane potential is represented as V. And then just talking about whatever the transmembrane potential is, it would be V sub Tm. If it's the resting transmembrane potential, it's V sub R. But V sub Tm is just the general transmembrane potential. It doesn't have to be resting or not. Okay? That potential is a consequence of two things. First of all, the fact that there's all of these proteins, acidic proteins, in the cell that by virtue of the redox potential in the cell are in their conjugate base form, and as such they're all anions. They're anionically charged. So if all that redox stuff didn't really register with you, it just means that there's a bunch of negative charges on these proteins. And because proteins are big, they're trapped. They can't get out. In fact, a lot of them are on macromolecules, and they're stuck in the cell. They're stuck in the cell. So there's a sequestered negative charge in the cell. Couple with that a bunch of proteins embedded in the cell that are actively transporting positive charge outside of the cell. If you have proteins that are ATPases that are burning energy to export positive charge, then you're going to get this potential where there's a buildup of positive charge on the outside and the sequestered negative charge on the inside. That's a potential difference. All right? That's energy to do work. All right? Is anybody foggy at all? Don't be shy. This is the time to tell me. Okay, it's all still pretty easy. So that was an electrical gradient. We can have other kinds of gradients. There can be a chemical gradient. So for example, look at this. These are just concentration gradients. Concentration gradients are entropically driven. They're entropically driven. Does anybody not understand what I mean by that? Entropy is the relative disorder in a system. Systems tend towards disorder. And if you sequester, if you impose order by like sorting all of one type of one thing to one place versus another, then you're imposing some kind of pattern or order on the system that is opposing the nature's tendency towards increasing entropy. Okay, so there is energy in that can drive a process. This is what chemical gradients are. This is what diffusion is. So here, sodium, there's a lot more sodium on this side of this membrane than there is on this side. There's just a little bit. So sodium's chemical gradient is sending it in this direction. If you were to let sodium just do what it wanted, we have 1, 2, 3, 4, 5, 6 over here and only 2 over there. 2 of these sodium would want to hop over to that side, so there'd be 4 on this side and 4 on that side. You say, well, that seems magical. That seems magical. How does that happen? Yeah, well, imagine each of these sodium atoms on either side have equal likelihood of wanting to go to the other side, right? There's an equal chance of any of them wanting to go over. But because there's so many of them over here, there's so many of them, there's a higher likelihood that one of these guys is going to go this way than one of these two is going to go that way in the same span of time. And with time, it will equilibrate so that the 4 on this side, so that the probability, thank you, of the probability of a sodium passing from this way to that is the same as that way to this. Okay? And it's the same with all these. I could go through the rest of the colors, but you can see how the numbers match up with the chemical gradients that they're facing, I think. All right? Is anyone not clear on that? So I covered electrical gradient and I covered chemical gradient. And now I'm going to put them together and show how the two of them work in concert. They work together. All right. First, before we perturb the membrane potential, let's talk about the resting potential. All right, so it turns out that the resting transmembrane potential in most cells, not all, there are some cells that are a little bit different, but they're not that different. They're all about negative 70 millivolts. Negative 70 millivolts. That's a pretty good number to put in your head. Negative 70 millivolts. And how does the cell maintain this? Well, it's kind of interesting. The cell, first of all, has these leak channels. There's a potassium leak channel. There's a sodium leak channel. The ions that are primarily responsible for maintaining the transmembrane potential are cations, first of all. Given what I've already told you today, why is it cations that we're concerned with here? What did I say about a cell early on, just a few slides ago when I was talking about electrical gradients? Yeah, the negative charge is stuck in here on the proteins, all right? So it's the motion of positive charge, which is going to give us the membrane potential. And those cations are potassium and sodium. And we'll come to understand why we need two of them and not just one in a moment. All right? But I'll give you the short answer now, and I'll rationalize it later. One of them, sodium, is going to be the trigger for destabilizing the membrane, and potassium is going to be the recovery ion that brings us back to transmembrane. So we need two of them. Okay? And it turns out that because they have opposite roles, they're going to have opposite gradients driving them. All right? So we have leak channels for potassium and sodium. And we have, and that means that potassium is able to pass through this channel in either direction. But it turns out that and we'll see why in a moment, but potassium's gradient is heading from the inside of the cell to the outside of the cell. Whoa. Why is that? Well, we'll find out in a bit. But it's a little bit counterintuitive, though, hey? The potassium, the positive, you might imagine it wants to be near this negative charge. But its gradient is pushing it out. We'll find out why in a moment. And then there is the sodium leak channel. And sodium, its gradient pulls it inside. Now, if that was all there was, these leak channels, then soon that membrane would come to a point of equilibrium. Not steady state, equilibrium. And I'm going to explain the difference between those two words in a moment. If you can appreciate the difference between steady state and equilibrium, you'll be one up on most of the science majors here at Colby, I think. But the sodium-potassium exchange pump is the answer. The sodium-potassium ATPase. It's an ATPase. It burns energy. Our cell, it's called constitutively on. It's constitutively active. Constitutively means all the time. That's all that word means. Constitutively active. So the sodium-potassium exchange pump, it is an antiporter. It's an antiporter. It burns one ATP, turns it into ADP, cleaves that phosphodiester bond, and in so doing, it drives two potassiums inside the cell and up the concentration gradient for potassium, and three sodium outside the cell and up sodium's concentration gradient. You guys getting this? All right. So there is a net, for every ATP we burn, there is a net increase in positive charge outside the cell of +1. That is what establishes the membrane potential. And the precise transmembrane potential is in part a function of the expression patterns of leak channels and the sodium-potassium ATPase. How many of each of those proteins is in the membrane is in part going to dictate exactly what that membrane potential is. You guys getting this? Is this new stuff yet? Am I onto new stuff for y'all? This is all old? Okay, sorry. Well, we'll get to some newer stuff here in a moment. It's hard to know. I guess I should sit through a 163, 64 class. No, I don't want to. I agree. Probably not any room in the classroom for me. They're so packed. All right. So if you can appreciate, I'm sure not everybody is familiar with this. So if you can appreciate this slide, this is probably one of the most important slides in the day. And what I'm going to do here is describe what the electrochemical gradient is. The combined electrical and chemical gradient for potassium and for sodium. So here we are at resting potential on this side. All right? And let's consider the top, the potassium. So the potassium's chemical gradient wants it to go outside of the cell, right? And that's because of the sodium-potassium ATPase. It's been pumping all this potassium inside the cell. So the chemical gradient wants it to go outside. But the electrical gradient wants it to go inside. It wants it to go inside. The electrical gradient is smaller than the chemical gradient. The electrical gradient wants it to go inside because there is a net positive charge on the outside. Even though there's all this potassium inside, there's more sodium on the outside because I pumped 3 sodium out for every 2 potassium I brought in. So there's a buildup of positive charge out there in the white. Yeah.
But is chemical gradient only?
Yeah. The chemical gradient is specific to potassium. The electrical gradient is specific to any cation. Okay? And so the net electrochemical gradient is net outside. It wants to go outside. Now, let's consider sodium. The sodium chemical gradient is inside, wants it to head inside, inward, influx. And that's because of what? What's making it such that there's a net drive towards influx of sodium? Huh? Say it. Thank you. The sodium-potassium ATPase. And that adds to the same electrical gradient that potassium is feeling. They're cations. They're both cations. So these two add up for there to be a large, a potent, a powerful tendency for influx of sodium. The electrochemical gradient, the electrical and the chemical gradient are additive. here. So there's a strong tendency for sodium to want to come inside. That's a pretty good looking trigger right there. You want to let that sodium go. It'll start ripping inside the cell. Strong, okay? So let's do it. Let's do them one at a time. Let's make the membrane permeable to potassium, but not sodium. Let's make it permeable to potassium, not sodium. So what's going to happen is a little bit of potassium is going to go outside the cell until the chemical gradient is equal and opposite to the electrical gradient. All right? If they're equal and opposite in force, then they're going to cancel, and there's going to be no net efflux or influx of potassium. Yes? No? Sort of? You're taking me at my word? So let's just open up a channel, not for sodium, just for potassium. We're just going to let potassium flow out. Some potassium is going to flow out to the point where enough has flowed out so that the chemical gradient is going to be equal and opposite to the electrical gradient. All right? So at that point, the forces driving it in are equal to the forces driving it out. And so that is equilibrium. That's an equilibrium. Let me define equilibrium and steady state, by the way. Equilibrium is any situation in which there's two states that are the chance of going in one direction is in balance with the chance of going in the other direction without any active input. All right, there's not active input any longer. Steady state is a system where the forward reaction and the backward reaction are not changing, but they're not changing because you are putting energy into the system. All right. Steady state. If your body came to equilibrium, you would be dead. That's equivalent to metabolic death. Right. So here, but that's not what the cell doesn't. I don't mean that the cell is dead. I'm just saying that if we were to release potassium to do what it wants, it's going to come to this resting potential. Its equilibrium potential for potassium is minus 90. It's minus 90. All right. So we have, yeah, sure. A steady state is in a situation in which the forward and back reactions between two states, okay, and it can be a chemical reaction, it can be any kind of dynamic transformation. A steady state is where the rate of the forward and the rate of the backward reactions are not changing due to an input of energy. All right, it's a steady state. It's not, the rates are not changing. Equilibrium, they're not changing either, but you're not putting energy in it. All right, you're letting the system equilibrate without any input. All right, steady state, eventually the battery is going to run out, and a steady state will dissipate and come to equilibrium. Does that make sense? Okay, that's the difference between those two terms. So most of life is a steady state. And in fact, these are really steady states, aren't they? I'm calling them resting potentials that come to equilibrium, but they're steady states because we have the sodium-potassium ATPase doing what it does. So let's consider sodium. We're going to make the membrane permeable to sodium, and what's going to happen? Sodium's going to rip inside the cell, and it's going to come in to such a degree that it's actually going to flip. It's going to flip the membrane potential. It's going to flip the membrane potential. So the membrane potential is going to become positive. It's going to go all the way from -70 all the way up past 0 to a plus 60 millivolts. It's going to flip that electrical gradient until the electrical gradient is equal and opposite to the chemical gradient. All right. So what we have here is one ion whose resting potential is negative, and the other whose equilibrium potential is positive, opposite directions. This is all because of the antiporter, the sodium-potassium ATPase. All right, so now I gave you a profoundly simplistic view of it. And a lot of times students imagine that like huge amounts of ions are sloshing here and there. And actually the real perturbations and concentrations of sodium and potassium are quite small. And the real way to determine transmembrane potential, the VTM, is the Goldman-Hodgkin-Katz voltage equation. So I'm going to need you guys all to memorize this and be able to use this on a quiz for tomorrow morning. I'm just showing it to you so you see it, so that you can appreciate that, that it is a more nuanced and sophisticated process than I just presented to you. And if you actually want to use that equation, you can take Dr. Martin's class because he does. All right. So yeah, I guess I don't even need this slide. I've already kind of said all this. This is redundant. Let me skip that slide. This is redundant too. I should have cut these out. Yeah. Well, I'll just say this. This is again redundant, but it's maybe a little bit of redundancy helps here. So these voltage-gated channels that I talked about earlier, we're going to find them in neural axons. So the axon of a neuron. We're going to find them in skeletal muscles. We're going to find them in a place called the sarcolemma. We're going to talk about sarcolemma tomorrow morning. It's basically the endoplasmic reticulum. No. It's the plasma lemma, the plasma membrane of a muscle cell. And you're going to find it in cardiac muscle. All right? So those are the places you'll find it. And again, I want to reiterate the fact that some of these voltage-gated channels have activation gates and deactivation gates. All right? So there's one potential that'll open the channel and another potential that'll shut the channel, which is going to be super convenient when we finally get into what these graded and action potentials are. Who here has had action potentials and graded potentials before? Oh, okay. I don't know. Great. So a couple terms. Depolarization is transmembrane, is a decrease in VTM. Hyperpolarization is an increase in VTM. So for example, what permeability to what ion would lead to hyperpolarization? Potassium, thank you. And then permeability to what other? Yeah, sodium. Okay, good job, Grace. All right, on to graded potentials. So they're local. They don't spread far from the site of stimulation. Any stimulus that opens a gated channel is going to create a graded potential. All right? Anything that is going to open up a gate is going to give us a graded potential. Is it a chemical? Is it electrical? Is it mechanical? So let's go through the steps let's go through the steps. Jesus. And a graded potential. Come on. one at a time. So there's some kind of stimulus. The stimulus I'm showing here is chemical. There's some sort of chemically triggered opening of the channel. And the cations come flooding into the cell. It's going to take us away from minus 70. Because what did we say? You guys remember? What was our equilibrium potential for sodium? Positive 66. Good job. Positive 66. Yeah. So resting potential is minus 70. Equilibrium for potassium is minus 90. And sodium is plus 66. So we're going to start heading. We've opened up this sodium channel, and sodium's going to start coming in. We're going to start heading towards plus 66. So we start depolarizing the cell. And as we depolarize the cell, we get these local currents. Cation, sodium starts flowing in and diffusing out from the site of perturbation and changing the membrane potential along the way. As that happens, we get a spread in, we get a gradient in the transmembrane potential. Here, at the site of the stimulation, it's minus 60. Now it's minus 65 over here. And further away, it's still at resting potential. All right, that's a graded potential. So first, let's look at depolarization sodium. This is a shift towards 0. So if you imagine here is resting potential at minus 70. And here is increasing potential this way. We're going to, here, this is the membrane. And we're going to stimulate the membrane right at that spot. There's going to be, we're going to head towards minus or plus 66 or 0 or whatever. And it's going to be less the further away from where you get that stimulation. So right where the red is, maybe that's where some acetylcholine fell onto the membrane because there is a synaptic bouton there, synaptic knob. On the other hand, with potassium, site of stimulation is in the same spot, but it's going to change the membrane potential in the opposite direction. Okay, it's going to increase the negativity of the resting potential. It's going to head towards minus 90. All right. Is there anything else there? No. Okay. So these graded potentials can either be depolarizing or hyperpolarizing, depending upon what type of channel was opened. All right. It's changed. The transmembrane potential is changed most at the site of stimulus, and the effect is going to spread passively due to these local currents. All right. So you're going to stimulate it here, and it's going to spread out passively. The stronger the stimulus, the greater the change in the transmembrane potential. That's going to become important later because I'm going to talk about AM versus FM, and I'm not talking about radio. I'm talking about amplitude modulated information encoding versus frequency modulated. All right. Graded potentials are amplitude modulation. How intense of a signal, the amplitude of the signal is how we encode information with graded potentials. All right, on to the action potential, the almighty action potential. Large, rapid, reversible, propagated decrease in transmembrane potential. So what happens here is once you trigger an action potential, it spreads to it spreads equally to the entire excitable membrane. Now, this requires special gates. It requires special gates in electrical electrically triggered gates in that membrane. An action potential is going to link graded potentials that happen at the cell body, right? So maybe we have these dendrites that have all these graded potentials on them, and they're adding up to an electrical field inside the neuron that's going to trigger something at the axon hillock. So an action potential links a certain threshold of electrical field density at the cell body in the perikaryon to synaptic activity that's going to happen at the axon terminal, at the terminus here where the telodendria are. All right, that's all that an action potential is. It's just, honestly, all the real action happens in here, the integration of all of these graded potentials that are coming in. All this information comes in, and they add and subtract and whatever, and eventually we get to a threshold, a state, where the cell has deemed this state is actionable. This is an actionable state, at which point we start an action potential, and we just send a signal to the next neuron down the chain. Yeah? So a cell, an excitable cell can have an action potential. Inexcitable cells do not. All cells have a transmembrane potential. There's a transmembrane potential of some sort to all cells. All cells are going to have a transmembrane potential, but these excitable cells are able to have action potentials. All right, so let's go through the steps of an action potential. In the beginning, we have a cell that's at minus 70 resting potential, and we have two types of gates. One gate is permeable to sodium, and one gate is permeable to potassium. All right, so now we're at the axon hillock, and we're going to have some local currents from the various graded potentials in the cell, and they're going to cause this voltage-gated channel, this sodium channel, it gets activated. The activation gate, we get to a potential that triggers that activation gate. It's going to swing open. And then sodium is going to begin to start flooding in. The all or none principle. You don't have like kind of an action potential or like a weak one or a strong one. You have an action potential or you do not. You either reach the threshold and open that gate and start the dominoes, or you do not. Action potentials are like dominoes. Once you flick them, they go. Right? And you can't get dominoes to fall harder or softer than they do. They just fall over, and that's what happens with an action potential. Yes, Tanisha. What did I say? No, action potentials are voltage gated. Yeah, they end in the degranulation of neurotransmitters, which and sometimes there can be electrical synapses, but at the synapse, that is it's going to attach to a chemically gated channel on the postsynaptic neuron. But action potentials are electrically gated channels. Axon hillock. Good job. That's it. All right. All right. So next step, sodium comes ripping in the cell. And because there's that giant chemical gradient driving it inside and electrical gradient driving it inside. And so it's going to go down its electrochemical gradient. And in so doing, it's going to head towards, the transmembrane potential is going to head towards the the sodium equilibrium potential. It's going to head towards plus 66. It's not going to get there. It's going to head there. As it passes plus 30, a couple things are going to happen. First of all, this door, the deactivation gate on the sodium channel, when it gets to plus 30, is going to slam shut. And now, since we're at plus 30 millivolts, what did I say the equilibrium potential for potassium was? Minus 90. We're a long way from home, aren't we? We're a long way from home. Potassium, the gate opens up at plus 30, whereas this one closes. The potassium one opens up. We're going to now reset the membrane potential. Potassium is going to start ripping out of the cell. We're going to head towards minus 90. These gates are going to reset in the sodium, and when we get up to minus 90, this gate is going to shut. Now, we are at a more or less normal electrical potential, transmembrane potential. Not exactly. We're a little bit far away from it, like 20 millivolts negative. We'll get back to normal here in a bit. But our chemical gradient's a little thrown off. So there's going to be a period called the refractory period, I'll talk about in a moment, where we're going to reestablish the proper chemical gradient based upon the sodium-potassium ATPase. All right, that's going to help reestablish the chemical gradient. The refractory period. So here's this chart. This is time. This is potential. Here we go. This at minus 60, this is the threshold at which the action potential starts when the sodium channel swings open. Gradient potentials impinge upon the axon hillock, and the potential of the membrane reaches minus 60, channel swings open, sodium rushes in, we get to plus 30, sodium channel closes, potassium channel opens, and we start to repolarize. All channels close, the way I had shown, and then the ATPase brings us back to normal. So in this period, in this time period here, this is the absolute refractory period. Once you've started this, you cannot start another action potential. You have to let the action potential finish before you initiate another one. Whereas in a graded potential, say I dropped some acetylcholine on there and I'm depolarizing the cell, I can continue to depolarize it more by dropping more acetylcholine on it until those receptors are saturated or whatever. But there's like a gradation of how much you can do. Action potentials, you're not. It's like if you want to use the domino model, during this, you start knocking over the dominoes. You can't knock the dominoes over again while the dominoes are running, right? They're being knocked over. You've started the action potential, okay? And so this is the absolute refractory period. You can definitely not restart an action potential during this. The relative refractory period, if there is a really strong graded potential at the axon hillock, you could maybe start another action potential. Maybe. It's like if you had a really long set of dominoes, you knock it down, and the dominoes are way over towards the other end, and you started setting up the dominoes again behind it, and you want to knock them down, so there's two waves of dominoes going, right? The second wave has started before the first wave is finished. But that's pretty hard to do, right? You have to really be into dominoes to do that. All right. Got it? Is this all still well-traveled ground? Okay. So here's this temporal profile that I just showed you. Here's the temporal profile. Here is that same thing. I'm sorry, it's such a crappy image that I have. I should just make one instead of some low-res thing off Google image. But this is a spatial profile. A spatial profile. If you imagine here is potential, And instead of time, this is just distance from the axon hillock. All right, here's the axon hillock. Here would be the synapse. All right, and this would be the propagation of that action potential across that membrane. Does that make sense? Here at the leading edge, these sodium channels are just starting to swing open. These sodium channels in this place on the membrane are slamming shut and potassium is opening. And here we're setting it up again. All right? So that's the spatial profile of the same thing. Here's just the same thing in a different picture. Okay. I don't want to say anything here. So this is just showing, this is the absolute refractory period. This would be maybe the relative refractory period. And that, you can see the perturbation in the membrane potential, and that's getting propagated along the axon towards that, towards the arborization at the synapse. So there's two methods of propagating action potentials. There's continuous propagation. Only a few neurons in humans or even chordates are continuously propagated. Most of them are what are called saltatory. But these are just continuously sweeping along the length of the axon. Now, the thing about continuous propagation is it requires a large diameter of the cell. Oh, we'll get to that actually in a moment. I think I'll talk about that in a moment. Let's just go through the steps of continuous propagation. So we have a gradient potential that triggers an action potential here at the axon hillock. It's depolarized. The membrane, the sodium has rushed in. We're at plus 30. But the leading edge of the axon is still at minus 70. So there's going to be a local current that just continuously sweeps along the length of the axon. And to the point where this portion of the membrane reaches threshold and begins to depolarize. Sodium continues this local current just sweeping along the length of the axon. Sodium is rushing in. We're in the refractory period here where we had just gone through the action potential, and local currents continue. So this is unidirectional. It can't go backwards because of the refractory period buffer behind it. All right? It doesn't, like once the action potential starts going along the membrane, it can't once membrane has reestablished itself behind it, it doesn't travel backwards again because that refractory period is a spatial buffer that makes it unidirectional. Is that making sense? Does that seem to make sense? So it travels in one direction about 1 meter per second. Kind of slow, actually. Kind of slow. Seems like that would be fast, but no, it's not. The next is saltatory propagation. So saltatory propagation happens in myelinated axons. A myelinated axon is an axon that has this fatty cell, either a Schwann cell or an oligodendrocyte, whether it's in the periphery or in the central nervous system. And these fatty cells act as insulators, keeping the electric field confined to the cytoplasm. So without getting into the physics too deeply, which would be a little bit deep water for me, but there is an electric field as a current is passing along a line. There's an electric field that's generated. There's the right-hand rule. Who's taken physics yet in here? You guys remember that then? There's an electric field that is orthogonal to the axis of the current flow. Putting an insulator on it confines that electric field to the cytoplasm. All right. And so what's nice about this is that you can have, say this is the axon hillock, we have the stimulus of an action potential here. We have the influx. Instead of needing to have all of those, all of this membrane embedded with excitable proteins and triggered and local currents and blah, 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 this whole physical thing, this myelination here concentrates the electric field that's being generated here in this membrane and is able to have a direct electrical transfer from this node to this node. So there are these nodes that happen. And the internodes are where we have the myelination. The length of the myelination is dictated by the biophysics of the system and the magnitude of the electric field that's being generated at each of the nodes. Does that make sense? Is anybody silently despairing? Is everybody good? Keep smiling. So the local current jumps from node to node. It's much faster. It's much faster, uses less energy than continuous propagation, and in fact allows for, as you'll see in a moment, I'm going to put another equation up to thrill you. It makes the cross-sectional diameter of the axon much smaller. All right, so let's see how this works, step at a time. Depolarization at the first segment, local current, electric field gets transported to the next node. We reach the threshold at that node. Influx there, electrical field quickly propagates to the next, and so forth. All right. So I said that the reason we do this is it's more energy efficient, it's faster, and it gives us an advantage in terms of axon diameter. Let's look at that a little bit. So here is the relationship that dictates the velocity of transfer to a number of metrics, specifically the axon diameter, the membrane capacitance. All right, so capacitance is the ability of something to hold electrical charge. All right, how much electrical charge can be stored in an object? It's related to the dielectric constant of a substance, and it turns out that fat has a high dielectric. So also related to membrane resistance and axonal resistance. So if we look at this relationship, the velocity is proportional, not exactly equal to, but it is proportional to the inverse of the membrane capacitance. So what does that mean? That means as membrane capacitance goes up, that the velocity is going to go down. So a fatty membrane, a membrane that is highly insulated, is going to have less capacitance. All right, we're going to. That's one of the ways that myelination. That is the way that myelination helps. as membrane resistance goes up, the velocity goes down as well. Axonal diameter. The larger the axonal diameter, the higher the velocity. So we're trading off membrane capacitance versus axonal diameter is what's happening in saltatory versus continuous. This means that continuous propagation makes up for the losses of not having myelination by being thicker. If you go into a neuro lab, probably not this one, he doesn't use giant squid. I don't know what he uses. They use crayfish. Crayfish probably are unmyelinated as well, but the classic one was giant squid because there are these enormous, actually quite visible neurons that had very thick axonal diameters and really easy to work with experimentally, so that the squid was able to have adequate speeds along its neurons without the myelination. I don't need you to memorize this at all, but I want you to know that there's a trade-off between speed and size. All right, the larger the diameter, the lower the resistance. There's a trade-off between speed and the axon diameter. So you have to quadruple. I said that myelin insulation is a great way to reduce the membrane capacitance, and you have to quadruple the diameter to double the velocity. So myelin is a directly or indirectly proportional, I guess linearly proportional, and this is proportional as the square. So you have to quadruple this diameter, and that's why those squid axons are so enormous. All right, so here's some implications of the all or none principle. Of course, all or none means you've either triggered the action potential or not. You don't knock over some of the as long as the dominoes have been set up properly, you don't knock over some of them and not the others. And an action potential, because of this all or none principle, is binary. It's you can think of it as digital information encoding. You either send a signal or there's not a signal. All right. And the informational content in the action potential is frequency encoded. So let's compare these. These would be signals for an FM. Let's think about it as radio for a moment. I know most of you probably don't listen to the AM radio, but there is in most cars still an AM radio band. And this is how the signal in an AM radio gets sent. There is a carrier frequency, whatever that frequency is, 10, 20 kilohertz or something like that. And that is this frequency that you see here. And that's the carrier frequency, but embedded upon that, on the amplitude of the signal, is another frequency. So you see this here. This frequency that you see of this swelling and shrinking of the amplitude, the height of those peaks, that is the audio signal getting imposed on top of the carrier signal. Is this familiar to everybody before? The whole notion of amplitude modulation? Okay, so a graded potential is just like this. The informational content in a graded potential is the amplitude or the strength, how intense was the stimulus that was put on the membrane. Whereas with an action potential, the informational content, since it's all or nothing, you're either sending a signal or you're not. And if that neuron is going to be sending a really it wants to increase the intensity of its signal, it needs to increase the rapidity at which it's sending action potentials. Does that make sense? So with a radio signal, for example, you have some sort of carrier frequency. Perhaps it's here. Maybe this is the carrier frequency. And instead of changing the amplitude, everything's the same height, but you're slightly changing the frequency. You're slightly changing that frequency, and you're embedding the frequency of the audio signal on top of the frequency of the carrier. You're changing the frequency of the carrier signal rather than the amplitude. So in terms of a neuron, if like the radio thing is confusing you, is it confusing anybody? No? Okay. In terms of comparing a graded potential and an action potential, a graded potential is sort of analog. It's like the comparison between analog and digital. It has a gradient of strength, whereas an action potential is binary and it's the rapidity of signaling. In fact, there's more than just the rapidity of it. We'll cover that in a bit. Not today, probably. Are there any questions thus far? All right. So here is kind of a recap of everything I've talked about today. I'm glad that this was review for some of you. Comparing graded potentials and action potentials. So graded potentials are either depolarizing or hyperpolarizing. They can either be it can either be something that opens a potassium channel and have it be hyperpolarizing, or something that opens a sodium channel and be depolarizing. There is no threshold, right? There's no threshold. It's just they're typically chemically or maybe mechanically gated. You just open some up, you cause a local current, and it dissipates with distance from the stimulus. The intensity depends upon the intensity of the stimulus. It passively spreads from the site of stimulus. It's not being driven by any kind of active mechanisms, any electrically gated triggers along the way. And there is no refractory period. We find this in most plasma membranes in the body. Most plasma membranes can have a graded potential of some sort. All cells have some kind of transmembrane potential. But excitable tissues are ones that can have action potentials. So an action potential is always going to be depolarizing. You have to hit a threshold before that happens. And it's all or none. You either start it or it hasn't. You don't reach the threshold. Once you reach the threshold, it's just like a snap trap, a mouse trap. Once it's been triggered, it goes. It doesn't go halfway. Once you reach threshold at one site along an excitable membrane, adjacent sites are going to be triggered until all of the excitable membrane has been triggered. It gets propagated along the entire length without any loss of strength in the signal. And there is a refractory period for a number of reasons. But one of the consequences of the refractory period is that it prevents a excitable membrane from going through continuous and not, that's the wrong word, it's a confusing word, through indefinite periods of depolarization. You can't have a backflow of the action potential. This only happens on excitable membrane. Excitable membrane is only in muscle and nerve tissue. All right. So even in nerve tissue, for example, the only excitable membrane that we have that can have action potentials, the only part of those cells is in the axon hillock and axon up to the synaptic boutons. The rest of it is not. It's just graded potentials. All right. So I cut out the information processing slides that I normally talk about because I was sensitive to slamming too much in your brain. But so what we can do now, we do have I ended a half hour, 25 minutes before I typically would have. I can do one of two things. We can go up to the lab now, and we can start the muscle lab, and we can go through the anatomy. I can give you a little bit of pre-lab, and then you'll have more time to just sit in the lab yourself. Or I can unhide the slides about information processing if you want to hear me talk about that a little bit. You want it to be a democracy. It is your education. What do you guys want? Who wants to go to the lab? Okay, it seems like most of you. I'll say this. Yeah, what's up? Question?
Where did dendrites?
The dendrites are little fingers that reach out from the cell body and have the synaptic boutons of other neurons attached to them. Okay. In sensory neurons, that they can be specialized and embedded in your skin or whatever they're doing. But the dendrites are where you're going to have a synapse from an upstream neuron.